Taxonomy, biology. In biology, taxonomy, from ancient Greek taxis, arrangement, and nomia, method, is the scientific study of naming, defining, circumscribing, and classifying groups of biological organisms based on shared characteristics. Organisms are grouped into taxa, singular, taxon, and these groups are given a taxonomic rank. Groups of a given rank can be aggregated to form a more inclusive group of higher rank, thus creating a taxonomic hierarchy. The principal ranks in modern use are domain, kingdom, phylum. Division is sometimes used in botany in place of phylum, class, order, family, genus, and species. The Swedish botanist Carl Linnaeus is regarded as the founder of the current system of taxonomy, as he developed a ranked system known as Linnaean taxonomy for categorizing organisms and binomial nomenclature for naming organisms. With advances in the theory, data and analytical technology of biological systematics, the Linnaean system has transformed into a system of modern biological classification intended to reflect the evolutionary relationships among organisms, both living and extinct. Definition. The exact definition of taxonomy varies from source to source, but the core of the discipline remains the conception, naming, and classification of groups of organisms. 1. As points of reference, recent definitions of taxonomy are presented below. 1. Theory and practice of grouping individuals into species, arranging species into larger groups, and giving those groups names, thus producing a classification. 2. A field of science and a major component of systematics that encompasses description, identification, nomenclature, and classification. 3. The science of classification, in biology the arrangement of organisms into a classification. 4. The science of classification is applied to living organisms, including the study of means of formation of species, etc. 5. The analysis of an organism's characteristics for the purpose of classification. 6. Systematics studies phylogeny to provide a pattern that can be translated into the classification and names of the more inclusive field of taxonomy, listed as a desirable but unusual definition. 7. The varied definitions either place taxonomy as a sub-area of systematics, definition 2, invert that relationship, definition 6, or appear to consider the two terms synonymous. There is some disagreement as to whether biological nomenclature is considered a part of taxonomy, definitions 1 and 2, or a part of systematics outside taxonomy. 8-9 For example, definition 6 is paired with the following definition of systematics that places nomenclature outside taxonomy. 6. Systematics, the study of the identification, taxonomy, and nomenclature of organisms, including the classification of living things with regard to their natural relationships and the study of variation in the evolution of taxa. In 1970, Michener et al. Defined systematic biology and taxonomy, terms that are often confused and used interchangeably in relation to one another as follows. 10. Systematic biology, hereafter called simply systematics, is the field that a. Provides scientific names for organisms, b. Describes them, c. Preserves collections of them, d. Provides classifications for the organisms, keys for their identification, and data on their distributions, e. Investigates their evolutionary histories, and f. Considers their environmental adaptations. This is a field with a long history that in recent years has experienced a notable renaissance, principally with respect to theoretical content. Part of the theoretical material has to do with evolutionary areas, topics E and F above, the rest relates especially to the problem of classification. Taxonomy is that part of systematics concerned with topics A to D above. A whole set of terms including taxonomy, systematic biology, systematics, scientific classification, biological classification, and phylogenetics have at times had overlapping meanings, sometimes the same, sometimes slightly different, but always related and intersecting. 111 The broadest meaning of taxonomy is used here. The term itself was introduced in 1813 by de Candolle, in his Théorie élémentaire de la botanique. 12 John Lindley provided an early definition of systematics in 1830, although he wrote of systematic botany, rather than using the term systematics. 13 Europeans tend to use the terms systematics and biosystematics for the study of biodiversity as a whole, whereas North Americans tend to use taxonomy more frequently. 14 However, taxonomy, and in particular alpha taxonomy, is more specifically the identification, description, and naming, i.e., nomenclature of organisms. 15 While classification focuses on placing organisms within hierarchical groups that show their relationships to other organisms. Monograph and taxonomic revision. A taxonomic revision or taxonomic review is a novel analysis of the variation patterns in a particular taxon. This analysis may be executed on the basis of any combination of the various available kinds of characters, such as morphological, anatomical, palynological, biochemical, and genetic. A monograph or complete revision is a revision that is comprehensive for a taxon for the information given at a particular time, and for the entire world. Other partial revisions may be restricted in the sense that they may only use some of the available character sets or have a limited spatial scope. A revision results in a confirmation of or new insights in the relationships between the subtaxa within the taxon under study, which may lead to a change in the classification of these subtaxa, the identification of new subtaxa, or the merger of previous subtaxa. 16. Taxonomic characters. Taxonomic characters are the taxonomic attributes that can be used to provide the evidence from which relationships, the phylogeny, between taxa are inferred. 17. 18 kinds of taxonomic characters include 19. Morphological characters. General external morphology. Special structures e.g., genitalia. Internal morphology, anatomy. embryology, karyology and other cytological factors, physiological characters, metabolic factors, body secretions, genic sterility factors, molecular characters, immunological distance, electrophoretic differences, amino acid sequences of proteins, DNA hybridization, DNA and RNA sequences, restriction endonuclease analyses, other molecular differences, behavioral characters, courtship and other ethological isolating mechanisms, other behavior patterns, ecological characters, habit and habitats, food, seasonal variations, parasites and hosts, geographic characters, general biogeographic distribution patterns, sympatric-allopatric relationship of populations, alpha and beta taxonomy. The term, alpha taxonomy, is primarily used to refer to the discipline of finding, describing, and naming taxa, particularly species. 20. In earlier literature, the term had a different meaning, referring to morphological taxonomy, and the products of research through the end of the 19th century. 21. William Bertram Turrell introduced the term, alpha taxonomy, in a series of papers published in 1935 and 1937 in which he discussed the philosophy and possible future directions of the discipline of taxonomy. 22. There is an increasing desire amongst taxonomists to consider their problems from wider viewpoints, to investigate the possibilities of closer cooperation with their cytological, ecological and genetics colleagues, and to acknowledge that some revision or expansion, perhaps of a drastic nature, of their aims and methods, may be desirable. Turrell, 1935, has suggested that while accepting the older invaluable taxonomy, based on structure, and conveniently designated, alpha, it is possible to glimpse a far distant taxonomy built upon as wide a basis of morphological and physiological facts as possible, and one in which, place is found for all observational and experimental data relating, even if indirectly, to the constitution, subdivision, origin, and behavior of species and other taxonomic groups. Ideals can, it may be said, never be completely realized. They have, however, a great value of acting as permanent stimulants, and if we have some, even vague, ideal of an omega taxonomy, we may progress a little way down the Greek alphabet. Some of us please ourselves by thinking we are now groping in a beta taxonomy. 22. Turrell thus explicitly excludes from alpha taxonomy various areas of study that he includes within taxonomy as a whole, such as ecology, physiology, genetics, and cytology. He further excludes phylogenetic reconstruction from alpha taxonomy. 23. Later authors have used the term in a different sense, to mean the delimitation of species, not subspecies or taxa of other ranks, using whatever investigative techniques are available, and including sophisticated computational or laboratory techniques. 24. Thus, Ernstmeyer in 1968 defined beta taxonomy as the classification of ranks higher than species. 25. An understanding of the biological meaning of variation and of the evolutionary origin of groups of related species is even more important for the second stage of taxonomic activity, the sorting of species into groups of relatives, taxa, and their arrangement in a hierarchy of higher categories. This activity is what the term classification denotes. It is also referred to as beta taxonomy, microtaxonomy and macrotaxonomy. How species should be defined in a particular group of organisms gives rise to practical and theoretical problems that are referred to as the species problem. The scientific work of deciding how to define species has been called microtaxonomy. 26, 27, 20. By extension, macrotaxonomy is the study of groups at the higher taxonomic ranks, subgenus and above. 20. Or simply enclaves that include more than one taxon considered a species, expressed in terms of phylogenetic nomenclature. 28. History. While some descriptions of taxonomic history attempt to date taxonomy to ancient civilizations, a truly scientific attempt to classify organisms did not occur until the 18th century, with the possible exception of Aristotle, whose works hint at a taxonomy. 29, 30. Earlier works were primarily descriptive and focused on plants that were useful in agriculture or medicine. There are a number of stages in this scientific thinking. Early taxonomy was based on arbitrary criteria, the so-called artificial systems, including Linnaeus's system of sexual classification for plants. Linnaeus's 1735 classification of animals was entitled Systema Natura, the system of nature, implying that he, at least, believed that it was more than an artificial system. Later came systems based on a more complete consideration of the characteristics of taxa, referred to as natural systems, such as those of de Jussieu 1789, de Candolle 1813, and Bentham and Hooker 1862-1863. These classifications described empirical patterns and were pre-evolutionary in thinking. The publication of Charles Darwin's On the Origin of Species 1859 led to a new explanation for classifications, based on evolutionary relationships. This was the concept of phyletic systems, from 1883 onwards. This approach was typified by those of Eichler 1883 and Engler 1886-1892. The advent of cladistic methodology in the 1970s led to classifications based on the sole criterion of monophily, supported by the presence of synapomorphies. Since then, the evidentiary basis has been expanded with data from molecular genetics that for the most part complements traditional morphology. 31, 32, 33. Pre-Linnaean. Early taxonomists. Naming and classifying human surroundings likely began with the onset of language. Distinguishing poisonous plants from edible plants is integral to the survival of human communities. Medicinal plant illustrations show up in Egyptian wall paintings from circa 1500 BC, indicating that the uses of different species were understood and that a basic taxonomy was in place. 34. Ancient times. Description of rare animals, Xie Xingjian Qin 2, by Song Dynasty painter Huang Quan, 903–965. Organisms were first classified by Aristotle, Greece, 384–322 BC, during his stay on the island of Lesbos. 35, 36, 37, he classified beings by their parts, or in modern terms attributes, such as having live birth, having 4 legs, laying eggs, having blood, or being warm-bodied. 38, he divided all living things into 2 groups, plants and animals. 36. Some of his groups of animals, such as Anhaema, animals without blood, translated as invertebrates, and Enhaema, animals with blood, roughly the vertebrates as well as groups like the sharks and cetaceans, are commonly used. 39, 40, 41. His student Theophrastus, Greece, 370–285 BC, carried on this tradition, mentioning some 500 plants and their uses in his Historia Plantarum. Several plant genera can be traced back to Theophrastus, such as Cornus, Crocus, and Narcissus. 36. Medieval. Taxonomy in the Middle Ages was largely based on the Aristotelian system, 38, with additions concerning the philosophical and existential order of creatures. This included concepts such as the great chain of being in the Western scholastic tradition, 38, again deriving ultimately from Aristotle. The Aristotelian system did not classify plants or fungi, due to the lack of microscopes at the time, 37, as his ideas were based on arranging the complete world in a single continuum, as per the Scala Natura, the natural ladder. 36. This, as well, was taken into consideration in the great chain of being. 36. Advances were made by scholars such as Procopius, Timotheus of Gaza, Demetrios Papagomenos, and Thomas Aquinas. Medieval thinkers used abstract philosophical and logical categorizations more suited to abstract philosophy than to pragmatic taxonomy. 36. Renaissance and early modern. During the Renaissance and the Age of Enlightenment, categorizing organisms became more prevalent, 36, and taxonomic works became ambitious enough to replace the ancient texts. This is sometimes credited to the development of sophisticated optical lenses, which allow the morphology of organisms to be studied in much greater detail. One of the earliest authors to take advantage of this leap in technology was the Italian physician Andrea Cisalpino, 1519–1603, who has been called the first taxonomist. 42. His magnum opus De Plantis came out in 1583, and described more than 1,500 plant species. 43–44. 2 large plant families that he first recognized are in use, the Astraceae and Brassicaceae. 45. In the 17th century, John Ray, England, 1627–1705, wrote many important taxonomic works. 37. Arguably his greatest accomplishment was Methodus Plantarum Nova, 1682–46, in which he published details of over 18,000 plant species. At the time, his classifications were perhaps the most complex yet produced by any taxonomist, as he based his taxa on many combined characters. The next major taxonomic works were produced by Joseph Pétain de Tournefort, France, 1656–1708. 47. His work from 1700, Institutions Rei Herbarii, included more than 9,000 species in 698 genera, which directly influenced Linnaeus, as it was the text he used as a young student. 34. Linnaean era. Title page of Systema Naturae, Leiden, 1735. The Swedish botanist Carl Linnaeus, 1707–1778, 38, ushered in a new era of taxonomy. With his major works Systema Naturae first edition in 1735, 48, Species Plantarum in 1753, 49, and Systema Naturae 10th edition, 50, he revolutionized modern taxonomy. His works implemented a standardized binomial naming system for animal and plant species, 51, which proved to be an elegant solution to a chaotic and disorganized taxonomic literature. He not only introduced the standard of class, order, genus, and species, but also made it possible to identify plants and animals from his book, by using the smaller parts of the flower known as the Linnaean system. 51. Plant and animal taxonomists regard Linnaeus' work as a starting point for valid names at 1753 and 1758 respectively. 52. Names published before these dates are referred to as pre-Linnaean, and not considered valid with the exception of spiders published in Svenska Spindler, 53. Even taxonomic names published by Linnaeus himself before these dates are considered pre-Linnaean. 34. The digital era of taxonomy. Modern taxonomy is heavily influenced by technology such as DNA sequencing, bioinformatics, databases, and imaging. Modern system of classification. Evolution of the vertebrates at class level, width of spindles indicating number of families. Spindle diagrams are typical for evolutionary taxonomy. The same relationship, expressed as a cladogram typical for cladistics. A pattern of groups nested within groups was specified by Linnaeus' classifications of plants and animals, and these patterns began to be represented as dendrograms of the animal and plant kingdoms toward the end of the 18th century, well before Charles Darwin's On the Origin of Species was published. 37. The pattern of the natural system did not entail a generating process, such as evolution, but may have implied it, inspiring early transmutationist thinkers. Among early works exploring the idea of a transmutation of species were Zoonomia in 1796 by Erasmus Darwin, Charles Darwin's grandfather, and Jean-Baptiste Lamarck's Philosophie zoologique of 1809. 20. The idea was popularized in the Anglophone world by the speculative but widely read Vestiges of the Natural History of Creation, published anonymously by Robert Chambers in 1844. 5. With Darwin's theory, a general acceptance quickly appeared that a classification should reflect the Darwinian principle of common descent. 55. Tree of life representations became popular in scientific works, with known fossil groups incorporated. One of the first modern groups tied to fossil ancestors was birds. 56. Using the then newly discovered fossils of Archaeopteryx and Hesperornis, Thomas Henry Huxley pronounced that they had evolved from dinosaurs, a group formally named by Richard Owen in 1842. 57. 58. The resulting description, that of dinosaurs, giving rise to, or being, the ancestors of, birds, is the essential hallmark of evolutionary taxonomic thinking. As more and more fossil groups were found and recognized in the late 19th and early 20th centuries, paleontologists worked to understand the history of animals through the ages by linking together known groups. 59. With the modern evolutionary synthesis of the early 1940s, an essentially modern understanding of the evolution of the major groups was in place. As evolutionary taxonomy is based on Linnaean taxonomic ranks, the two terms are largely interchangeable in modern use. 60. The cladistic method has emerged since the 1960s. 55. In 1958, Julian Huxley used the term clade. 20. Later, in 1960, Kane and Harrison introduced the term cladistic. 20. The salient feature is arranging taxa in a hierarchical evolutionary tree, with the desideratum that all named taxa are monophyletic. 55. A taxon is called monophyletic if it includes all the descendants of an ancestral form. 61. 62. Groups that have descendant groups removed from them are termed paraphyletic. 61. While groups representing more than one branch from the tree of life are called polyphyletic. 61. 62. Monophyletic groups are recognized and diagnosed on the basis of synapomorphies, shared derived character states. 63. Cladistic classifications are compatible with traditional Linnaean taxonomy and the codes of zoological and botanical nomenclature, to a certain extent. 64. An alternative system of nomenclature, the International Code of Phylogenetic Nomenclature or Phylocode has been proposed, which regulates the formal naming of clades. 65. 28. 9. Linnaean ranks are optional and have no formal standing under the Phylocode, which is intended to coexist with the current, rank-based codes. 28. While popularity of phylogenetic nomenclature has grown steadily in the last few decades, 9. It remains to be seen whether a majority of systematists will eventually adopt the Phylocode or continue using the current systems of nomenclature that have been employed and modified, but arguably not as much as some systematists wished. 66. 67. For over 250 years. Kingdoms and domains. The basic scheme of modern classification. Many other levels can be used. Domain, the highest level within life, is both new and disputed. Well before Linnaeus, plants and animals were considered separate kingdoms. 68. Linnaeus used this as the top rank, dividing the physical world into the vegetable, animal and mineral kingdoms. As advances in microscopy made the classification of microorganisms possible, the number of kingdoms increased, 5 and 6 kingdom systems being the most common. Domains are a relatively new grouping. First proposed in 1977, Carl Woese's three-domain system was not generally accepted until later. 69. One main characteristic of the three-domain method is the separation of archaea and bacteria, previously grouped into the single-kingdom bacteria, a kingdom also sometimes called Monera. 68. With the eukaryota for all organisms whose cells contain a nucleus. 70. A small number of scientists include a sixth kingdom, archaea, but do not accept the domain method. 68. Thomas Cavalier Smith, who published extensively on the classification of protists, in 2002, 71, proposed that the Neomura, the clade that groups together the archaea and eukarya, would have evolved from bacteria, more precisely from Actinomycetota. His 2004 classification treated the Archaeobacteria as part of a subkingdom of the kingdom Bacteria, i.e., he rejected the three-domain system entirely. 72. Stefan Luketa in 2012 proposed a 5-dominion system, adding Prionobiota acellular and without nucleic acid, and Verusobiota acellular but with nucleic acid to the traditional three domains. 73. Linnaeus 1735, 74. Haeckel 1866, 75. Chatham 1925, 76. Copeland 1938, 77. Whittaker 1969, 78. Woese et al., 1990, 79. Cavalier Smith 1998, 72, 2015, 80. 2 kingdoms 3 kingdoms 2 empires 4 kingdoms 5 kingdoms 3 domains. 2 empires, 6/7 kingdoms. Not treated Protista. Prokaryota Monera Monera. Bacteria Bacteria Archaea Archaea, 20. Recent comprehensive classifications. Partial classifications exist for many individual groups of organisms and are revised and replaced as new information becomes available. However, comprehensive, published treatments of most or all life are rarer. Recent examples are that of Adel et al. 2012 and 2019, 81-82, which covers eukaryotes only with an emphasis on protists, and Ruggiero et al. 2015, 83, covering both eukaryotes and prokaryotes to rank of order, although both exclude fossil representatives. 83. A separate compilation, Ruggiero, 2014, 84, covers sextant taxa to the rank of family. Other, database-driven treatments include the Encyclopedia of Life, the Global Biodiversity Information Facility, the NCBI Taxonomy Database, the Interim Register of Marine and Non-Marine Genera, the Open Tree of Life, and the Catalog of Life. The Paleobiology Database is a resource for fossils. Application. Biological taxonomy is a subdiscipline of biology, and is generally practiced by biologists known as taxonomists, though enthusiastic naturalists are also frequently involved in the publication of new taxa. 85 Because taxonomy aims to describe and organize life, the work conducted by taxonomists is essential for the study of biodiversity and the resulting field of conservation biology. 86-87. Classifying organisms. Biological classification is a critical component of the taxonomic process. As a result, it informs the user as to what the relatives of the taxon are hypothesized to be. Biological classification uses taxonomic ranks, including among others in order from most inclusive to least inclusive domain, kingdom, phylum, class, order, family, genus, species, and strain. 88 Note 1. Taxonomic descriptions. Type specimen 4. Nepenthes smilesii, a tropical pitcher plant. The definition of a taxon is encapsulated by its description or its diagnosis or by both combined. There are no set rules governing the definition of taxa, but the naming and publication of new taxa is governed by sets of rules. 8 In zoology, the nomenclature for the more commonly used ranks, superfamily to subspecies, is regulated by the International Code of Zoological Nomenclature (ICZN Code). 89 In the fields of phycology, mycology, and botany, the naming of taxa is governed by the International Code of Nomenclature for Algae, Fungi, and Plants (ICN). 90. The initial description of a taxon involves 5 main requirements. 91. 1. The taxon must be given a name based on the 26 letters of the Latin alphabet, a binomial for new species, or uninomial for other ranks. 2. The name must be unique, i.e. not a homonym. 3. The description must be based on at least one name-bearing type specimen. 4. It should include statements about appropriate attributes either to describe, define, the taxon or to differentiate it from other taxa, the diagnosis, ICZN Code, Article 13.1.1, ICN, Article 38, which may or may not be based on morphology. 92. Both codes deliberately separate defining the content of a taxon, its circumscription, from defining its name. 5. These first 4 requirements must be published in a work that is obtainable in numerous identical copies, as a permanent scientific record. However, often much more information is included, like the geographic range of the taxon, ecological notes, chemistry, behavior, etc. How researchers arrive at their taxa varies, depending on the available data and resources. Methods vary from simple quantitative or qualitative comparisons of striking features, to elaborate computer analyses of large amounts of DNA sequence data. 93. Author citation. An authority may be placed after a scientific name. 94. The authority is the name of the scientist or scientists who first validly published the name. 94. For example, in 1758, Linnaeus gave the Asian elephant the scientific name Elephas maximus, so the name is sometimes written as Elephas maximus Linnaeus, 1758. 95. The names of authors are often abbreviated. The abbreviation L, for Linnaeus, is commonly used. In botany, there is, in fact, a regulated list of standard abbreviations, see list of botanists by author abbreviation. 96. The system for assigning authorities differs slightly between botany and zoology. 8. However, it is standard that if the genus of a species has been changed since the original description, the original authority's name is placed in parentheses. 97. Phonetics. A comparison of phylogenetic and phonetic character-based concepts. In phonetics, also known as taxometrics, or numerical taxonomy, organisms are classified based on overall similarity, regardless of their phylogeny or evolutionary relationships. 20. It results in a measure of hypergeometric distance between taxa. Phonetic methods have become relatively rare in modern times, largely superseded by cladistic analyses, as phonetic methods do not distinguish shared ancestral or plesiomorphic traits from shared derived or apomorphic traits. 98. However, certain phonetic methods, such as neighbor joining, have persisted, as rapid estimators of relationships when more advanced methods, such as Bayesian inference, are too computationally expensive. 99. Databases. Modern taxonomy uses database technologies to search and catalog classifications and their documentation. 100. While there is no commonly used database, there are comprehensive databases such as the Catalog of Life, which attempts to list every documented species. 101. The catalog listed 1.64 million species for all kingdoms as of April 2016, claiming coverage of more than 3/4 of the estimated species known to modern science. Cladistics. Cladistics Cladstiques KL dist IKS, from ancient Greek klados branch. 1 is an approach to biological classification in which organisms are categorized in groups clades, based on hypotheses of most recent common ancestry. The evidence for hypothesized relationships is typically shared derived characteristics, synapomorphies, that are not present in more distant groups and ancestors. However, from an empirical perspective, common ancestors are inferences based on a cladistic hypothesis of relationships of taxa whose character states can be observed. Theoretically, a last common ancestor and all its descendants constitute a minimal clade. Importantly, all descendants stay in their overarching ancestral clade. For example, if the terms worms or fishes were used within a strict cladistic framework, these terms would include humans. Many of these terms are normally used paraphyletically, outside of cladistics, e.g. as a grade, which are fruitless to precisely delineate, especially when including extinct species. Radiation results in the generation of new subclades by bifurcation, but in practice sexual hybridization may blur very closely related groupings. 2, 3, 4, 5. As a hypothesis, a clade can be rejected only if some groupings were explicitly excluded. It may then be found that the excluded group did actually descend from the last common ancestor of the group, and thus emerged within the group. Evolved from is misleading, because in cladistics all descendants stay in the ancestral group. To keep only valid clades, upon finding that the group is paraphyletic this way, either such excluded groups should be granted to the clade, or the group should be abolished. 6. Branches down to the divergence to the next significant, e.g. extant, sister are considered stem groupings of the clade, but in principle each level stands on its own, to be assigned a unique name. For a fully bifurcated tree, adding a group to a tree also adds an additional, named clade, and a new level on that branch. Specifically, also extinct groups are always put on a side branch, not distinguishing whether an actual ancestor of other groupings was found. The techniques and nomenclature of cladistics have been applied to disciplines other than biology. See phylogenetic nomenclature. Cladistics findings are posing a difficulty for taxonomy, where the rank and genus naming of established groupings may turn out to be inconsistent. Cladistics is now the most commonly used method to classify organisms. 7. History. Willi Hennig 1972. Peter Chalmers Mitchell in 1920. Robert John Tilliard. The original methods used in cladistic analysis in the school of taxonomy derived from the work of the German entomologist Willi Hennig, who referred to it as phylogenetic systematics, also the title of his 1966 book, but the terms cladistics and clade were popularized by other researchers. Cladistics in the original sense refers to a particular set of methods used in phylogenetic analysis, although it is now sometimes used to refer to the whole field. 8. What is now called the cladistic method appeared as early as 1901 with a work by Peter Chalmers Mitchell for birds, 9, 10, and subsequently by Robert John Tilliard for insects in 1921, 11, and W. Zimmerman for plants in 1943. 12. The term clade was introduced in 1958 by Julian Huxley after having been coined by Lucian Quenaut in 1940. 13. Cladogenesis, in 1958. 14. Cladistic, by Arthur Kane and Harrison in 1960. 15. Cladist, for an adherent of Hennig's school by Ernst Mayr in 1965. 16. And cladistics, in 1966. 14. Hennig referred to his own approaches, phylogenetic systematics. From the time of his original formulation until the end of the 1970s, cladistics competed as an analytical and philosophical approach to systematics with phonetics and so-called evolutionary taxonomy. Phonetics was championed at this time by the numerical taxonomists Peter Sneath and Robert Sokol, and evolutionary taxonomy by Ernst Mayr. 17. Originally conceived, if only in essence, by Willi Hennig in a book published in 1950, cladistics did not flourish until its translation into English in 1966, Lewin 1997. Today, cladistics is the most popular method for inferring phylogenetic trees from morphological data. In the 1990s, the development of effective polymerase chain reaction techniques allowed the application of cladistic methods to biochemical and molecular genetic traits of organisms, vastly expanding the amount of data available for phylogenetics. At the same time, cladistics rapidly became popular in evolutionary biology, because computers made it possible to process large quantities of data about organisms and their characteristics. Methodology. The cladistic method interprets each shared character state transformation as a potential piece of evidence for grouping. Synapomorphies, shared, derived character states, are viewed as evidence of grouping, while simpliciomorphies, shared ancestral character states, are not. The outcome of a cladistic analysis is a cladogram, a tree-shaped diagram, dendrogram, 18, that is interpreted to represent the best hypothesis of phylogenetic relationships. The outcome of a cladistic analysis is a cladogram, a tree-shaped diagram, dendrogram, 18, that is interpreted to represent the best hypothesis of phylogenetic relationships. Although traditionally such cladograms were generated largely on the basis of morphological characters and originally calculated by hand, genetic sequencing data and computational phylogenetics are now commonly used in phylogenetic analyses, and the parsimony criterion has been abandoned by many phylogeneticists in favor of more sophisticated, but less parsimonious evolutionary models of character state transformation. Cladists contend that these models are unjustified because there is no evidence that they recover more true or correct results from actual empirical datasets. 19. Every cladogram is based on a particular dataset analyzed with a particular method. Datasets are tables consisting of molecular, morphological, ethological 20, and or other characters in a list of operational taxonomic units (OTUS), which may be genes, individuals, populations, species, or larger taxa that are presumed to be monophyletic and therefore to form, altogether, one large clade. Phylogenetic analysis infers the branching pattern within that clade. Different datasets and different methods, not to mention violations of the mentioned assumptions, often result in different cladograms. Only scientific investigation can show which is more likely to be correct. For example, cladograms like the following have generally been accepted as accurate representations of the ancestral relations among turtles, lizards, crocodilians, and birds. 21. Filled down pointing triangle. Testudinous turtles. Dipsida diamond suit. Lepidosauria lizards. Archosauria. Crocodilomorpha crocodilians dinosauria birds. If this phylogenetic hypothesis is correct, then the last common ancestor of turtles and birds, at the branch near the filled down pointing triangle lived earlier than the last common ancestor of lizards and birds, near the diamond suit. Most molecular evidence, however, produces cladograms more like this. 22. Dipsida diamond suit. Lepidosauria lizards. Archosauromorpha filled down pointing triangle. Testudinous turtles. Archosauria. Crocodilomorpha crocodilians dinosauria birds. If this is accurate, then the last common ancestor of turtles and birds lived later than the last common ancestor of lizards and birds. Since the cladograms show two mutually exclusive hypotheses to describe the evolutionary history, at most one of them is correct. Cladogram of the primates, showing a monophyletic taxon, a clade, the simians or Anthropoidea, in yellow, a paraphyletic taxon, the prosimians, in blue, including the red patch, and a polyphyletic taxon, the nocturnal primates, the lorises and the tarsiers, in red. The cladogram to the right represents the current universally accepted hypothesis that all primates, including strepsiorhines like the lemurs and lorises, had a common ancestor all of whose descendants are or were primates, and so form a clade. The name primates is therefore recognized for this clade. Within the primates, all anthropoids, monkeys, apes, and humans are hypothesized to have had a common ancestor all of whose descendants are or were anthropoids, so they form the clade called Anthropoidea. The prosimians, on the other hand, form a paraphyletic taxon. The name prosimy is not used in phylogenetic nomenclature, which names only clades. The prosimians are instead divided between the clades Strepsiorhini and Haplorhini, where the latter contains tarsiforms and Anthropoidea. Lemurs and tarsiers may have looked closely related to humans, in the sense of being close on the evolutionary tree to humans. However, from the perspective of a tarsier, humans and lemurs would have looked close, in the exact same sense. Cladistics forces a neutral perspective, treating all branches, extant or extinct, in the same manner. It also forces one to try to make statements, and honestly take into account findings, about the exact historic relationships between the groups. Terminology for character states. The following terms, coined by Hennig, are used to identify shared or distinct character states among groups. 23, 24, 25. A plesiomorphy, close form, or ancestral state is a character state that a taxon has retained from its ancestors. When two or more taxa that are not nested within each other share a plesiomorphy, it is a symplesiomorphy, from syn, together. Symplesiomorphies do not mean that the taxa that exhibit that character state are necessarily closely related. For example, Reptilia is traditionally characterized by, among other things, being cold-blooded, i.e., not maintaining a constant high body temperature, whereas birds are warm-blooded. Since cold-bloodedness is a plesiomorphy, inherited from the common ancestor of traditional reptiles and birds, and thus a symplesiomorphy of turtles, snakes and crocodiles, among others, it does not mean that turtles, snakes and crocodiles form a clade that excludes the birds. An apomorphy, separate form, or derived state is an innovation. It can thus be used to diagnose a clade, or even to help define a clade name in phylogenetic nomenclature. Features that are derived in individual taxa, a single species or a group that is represented by a single terminal in a given phylogenetic analysis, are called autapomorphies, from auto, self. Features that are derived in individual taxa, a single species or a group that is represented by a single terminal in a given phylogenetic analysis, are called autapomorphies, from auto, self. Autapomorphies express nothing about relationships among groups. Clades are identified or defined by synapomorphies, from syn, together. For example, the possession of digits that are homologous with those of Homo sapiens is a synapomorphy within the vertebrates. The tetrapods can be singled out as consisting of the first vertebrate with such digits homologous to those of Homo sapiens together with all descendants of this vertebrate, an apomorphy-based phylogenetic definition. 26. Importantly, snakes and other tetrapods that do not have digits are nonetheless tetrapods. Other characters, such as amniotic eggs and diapsid skulls, indicate that they descended from ancestors that possess digits which are homologous with ours. A character state is homoplastic or, an instance of homoplasy, if it is shared by two or more organisms but is absent from their common ancestor or from a later ancestor in the lineage leading to one of the organisms. It is therefore inferred to have evolved by convergence or reversal. Both mammals and birds are able to maintain a high constant body temperature, i.e., they are warm-blooded. However, the accepted cladogram explaining their significant features indicates that their common ancestor is in a group lacking this character state, so the state must have evolved independently in the two clades. Warm-bloodedness is separately a synapomorphy of mammals, or a larger clade, and a birds, or a larger clade, but it is not a synapomorphy of any group including both these clades. Hennig's auxiliary principle 27 states that shared character states should be considered evidence of grouping unless they are contradicted by the weight of other evidence. Thus, homoplasy of some feature among members of a group may only be inferred after a phylogenetic hypothesis for that group has been established. The terms plesiomorphy and apomorphy are relative. Their application depends on the position of a group within a tree. For example, when trying to decide whether the tetrapods form a clade, an important question is whether having four limbs is a synapomorphy of the earliest taxa to be included within Tetrapoda. Did all the earliest members of the Tetrapoda inherit four limbs from a common ancestor, whereas all other vertebrates did not, or at least not homologously? By contrast, for a group within the tetrapods, such as birds, having four limbs is a plesiomorphy. Using these two terms allows a greater precision in the discussion of homology, in particular allowing clear expression of the hierarchical relationships among different homologous features. It can be difficult to decide whether a character state is in fact the same and thus can be classified as a synapomorphy, which may identify a monophyletic group, or whether it only appears to be the same and is thus a homoplasy, which cannot identify such a group. There is a danger of circular reasoning. Assumptions about the shape of a phylogenetic tree are used to justify decisions about character states, which are then used as evidence for the shape of the tree. 28. Phylogenetics uses various forms of parsimony to decide such questions. The conclusions reached often depend on the dataset and the methods. Such is the nature of empirical science, and for this reason, most cladists refer to their cladograms as hypotheses of relationship. Cladograms that are supported by a large number and variety of different A character state is homoplastic or an instance of homoplasy if it is shared by two or more organisms but is absent from their common ancestor or from a later ancestor in the lineage leading to one of the organisms. It is therefore inferred to have evolved by convergence or reversal. Both mammals and birds are able to maintain a high constant body temperature, i.e., they are warm-blooded. However, the accepted cladogram explaining their significant features indicates that their common ancestor is in a group lacking this character state, so the state must have evolved independently in the two clades. Warm-bloodedness is separately a synapomorphy of mammals, or a larger clade, and a birds, or a larger clade, but it is not a synapomorphy of any group including both these clades. Koenig's auxiliary principle 27 states that shared character states should be considered evidence of grouping unless they are contradicted by the weight of other evidence. Thus, homoplasy of some feature among members of a group may only be inferred after a phylogenetic hypothesis for that group has been established. The terms plesiomorphy and apomorphy are relative. Their application depends on the position of a group within a tree. For example, when trying to decide whether the tetrapods form a clade, an important question is whether having four limbs is a synapomorphy of the earliest taxa to be included within Tetrapoda. Did all the earliest members of the Tetrapoda inherit four limbs from a common ancestor, whereas all other vertebrates did not, or at least not homologously? By contrast, for a group within the tetrapods, such as birds, having four limbs is a plesiomorphy. Using these two terms allows a greater precision in the discussion of homology, in particular allowing clear expression of the hierarchical relationships among different homologous features. It can be difficult to decide whether a character state is in fact the same and thus can be classified as a synapomorphy, which may identify a monophyletic group, or whether it only appears to be the same and is thus a homoplasy, which cannot identify such a group. There is a danger of circular reasoning. Assumptions about the shape of a phylogenetic tree are used to justify decisions about character states, which are then used as evidence for the shape of the tree. 28. Phylogenetics uses various forms of parsimony to decide such questions. The conclusions reached often depend on the dataset and the methods. Such is the nature of empirical science, and for this reason, most cladists refer to their cladograms as hypotheses of relationship. Cladograms that are supported by a large number and variety of different kinds of characters are viewed as more robust than those based on more limited evidence. 29. Terminology for taxa. Mono, parent polyphyletic taxa can be understood based on the shape of the tree, as done above, as well as based on their character states. 24, 25, 30, these are compared in the table below. Term node-based definition, character-based definition. Holophily, monophyly. A clade, a monophyletic taxon, is a taxon that consists of the last common ancestor and all its descendants. 31. A clade is characterized by one or more apomorphies, derived character states present in the first member of the taxon, inherited by its descendants unless secondarily lost, and not inherited by any other taxa. Paraphily. A paraphyletic assemblage is one that is Holophily, monophyly. A clade, a monophyletic taxon, is a taxon that consists of the last common ancestor and all its descendants. 31. A clade is characterized by one or more apomorphies, derived character states present in the first member of the taxon, inherited by its descendants unless secondarily lost, and not inherited by any other taxa. Paraphily. A paraphyletic assemblage is one that is constructed by taking a clade and removing one or more smaller clades. 32. Removing one clade produces a singly paraphyletic assemblage, removing two produces a doubly paraphyletic assemblage, and so on. 33. A paraphyletic assemblage is characterized by one or more plesiomorphies, character states inherited from ancestors but not present in all of their descendants. As a consequence, a paraphyletic assemblage is truncated, in that it excludes one or more clades from an otherwise monophyletic taxon. An alternative name is evolutionary grade, referring to an ancestral character state within the group. While paraphyletic assemblages are popular among paleontologists and evolutionary taxonomists, cladists do not recognize paraphyletic assemblages as having any formal information content, they are merely parts of clades. Polyphily. A polyphyletic assemblage is one which is neither monophyletic nor paraphyletic. A polyphyletic assemblage is characterized by one or more homoplasies, character states which have converged or reverted so as to be the same but which have not been inherited from a common ancestor. No systematicist recognizes polyphyletic assemblages as taxonomically meaningful entities, although ecologists sometimes consider them meaningful labels for functional participants in ecological communities e.g., primary producers, detritivores, etc. Cladistics, either generally or in specific applications, has been criticized from its beginnings. Decisions as to whether particular character states are homologous, a precondition of their being synapomorphies, have been challenges involving circular reasoning and subjective judgments. 34. Of course, the potential unreliability of evidence is a problem for any systematic method, or for that matter, for any empirical scientific endeavor at all. 35. 36. Transformed cladistics arose in the late 1970s, 37, in an attempt to resolve some of these problems by removing a priori assumptions about phylogeny from cladistic analysis, but it has remained unpopular. 38. Criticism. Issues. Ancestors. The cladistic method does not identify fossil species as actual ancestors of a clade. 39. Instead, fossil taxa are identified as belonging to separate extinct branches. While a fossil species could be the actual ancestor of a clade, there is no way to know that. Therefore, a more conservative hypothesis is that the fossil taxon is related to other fossil and extant taxa, as implied by the pattern of shared apomorphic features. 40. Extinction status. An otherwise extinct group with any extant descendants is not considered literally extinct. 41, and for instance does not have a date of extinction. Hybridization, interbreeding. Anything having to do with biology and sex is complicated and messy, and cladistics is no exception. 42. Many species reproduce sexually, and are capable of interbreeding for millions of years. Worse, during such a period, many branches may have radiated, and it may take hundreds of millions of years for them to have whittled down to just 2. 43. Only then one can theoretically assign proper last common ancestors of groupings which do not inadvertently include earlier branches. 44. The process of true cladistic bifurcation can thus take a much more extended time than one is usually aware of. 45. In practice, for recent radiations, cladistically guided findings only give a coarse impression of the complexity. A more detailed account will give details about fractions of introgressions between groupings, and even geographic variations thereof. This has been used as an argument for the use of paraphyletic groupings. 44. But typically other reasons are quoted. Horizontal gene transfer. Horizontal gene transfer is the mobility of genetic info between different organisms that can have immediate or delayed effects for the reciprocal host. 46. There are several processes in nature which can cause horizontal gene transfer. This does typically not directly interfere with ancestry of the organism, but can complicate the determination of that ancestry. On another level, one can map the horizontal gene transfer processes by determining the phylogeny of the individual genes using cladistics. Naming stability. If there is unclarity in mutual relationships, there are a lot of possible trees. Assigning names to each possible clade may not be prudent. Furthermore, established names are discarded in cladistics, or alternatively carry connotations which may no longer hold, such as when additional groups are found to have emerged in them. 47. Naming changes are the direct result of changes in the recognition of mutual relationships, which often is still in flux, especially for extinct species. Hanging on to older naming and/or connotations is counterproductive, as they typically do not reflect actual mutual relationships precisely at all. E.g. Archaea, Asgard archaea, protists, slime molds, worms, invertebrata, fishes, reptilia, monkeys, Ardipithecus, Australopithecus, Homo erectus all contain Homo sapiens cladistically, in their sensu lato meaning. For originally extinct stem groups, sensu lato generally means generously keeping previously included groups, which then may come to include even living species. A prune sensu stricto meaning is often adopted instead, but the group would need to be restricted to a single branch on the stem. Other branches then get their own name and level. This is commensurate to the fact that more senior stem branches are in fact closer related to the resulting group than the more basal stem branches. That those stem branches only may have lived for a short time does not affect that assessment in cladistics. In disciplines other than biology, the comparisons used to acquire data on which cladograms can be based are not limited to the field of biology. 48. Any group of individuals or classes that are hypothesized to have a common ancestor, and to which a set of common characteristics may or may not apply, can be compared pairwise. Cladograms can be used to depict the hypothetical descent relationships within groups of items in many different academic realms. The only requirement is that the items have characteristics that can be identified and measured. Anthropology and archaeology. 49. Cladistic methods have been used to reconstruct the development of cultures or artifacts using groups of cultural traits or artifact features. Comparative mythology and folktale use cladistic methods to reconstruct the proto-version of many myths. Mythological phylogenies constructed with mythemes clearly support low horizontal transmissions, borrowings, historical, sometimes Paleolithic, diffusions and punctuated evolution. 50. They also are a powerful way to test hypotheses about cross-cultural relationships among folktales. 51. 52. Literature. Cladistic methods have been used in the classification of the surviving manuscripts of the Canterbury Tales, 53, and the manuscripts of the Sanskrit Charaka Samhita. 54. Historical linguistics. 55. Cladistic methods have been used to reconstruct the phylogeny of languages using linguistic features. This is similar to the traditional comparative method of historical linguistics, but is more explicit in its use of parsimony and allows much faster analysis of large datasets, computational phylogenetics. Textual criticism or stemmatics. 54. 56. Cladistic methods have been used to reconstruct the phylogeny of manuscripts of the same work, and reconstruct the lost original, using distinctive copying errors as apomorphies. This differs from traditional historical comparative linguistics in enabling the editor to evaluate and place in genetic relationship large groups of manuscripts with large numbers of variants that would be impossible to handle manually. It also enables parsimony analysis of contaminated traditions of transmission that would be impossible to evaluate manually in a reasonable period of time. Astrophysics. 57. Infers a history of relationships between galaxies to create branching diagram hypotheses of galaxy diversification. Phonetics. In biology, phonetics phenx from ancient Greek phenein, to appear, also known as taxometrics, is an attempt to classify organisms based on overall similarity, usually with respect to morphology or other observable traits, regardless of their phylogeny or evolutionary relation. It is related closely to numerical taxonomy which is concerned with the use of numerical methods for taxonomic classification. Many people contributed to the development of phonetics, but the most influential were Peter Sneath and Robert R. Sokol. Their books are still primary references for this sub-discipline, although now out of print. 1. Phonetics has been largely superseded by cladistics for research into evolutionary relationships among species. However, certain phonetic methods, such as neighbor joining, are used for phylogenetics, as a reasonable approximation of phylogeny when more advanced methods, such as Bayesian inference, are too expensive computationally. Phonetic techniques include various forms of clustering and ordination. These are sophisticated methods of reducing the variation displayed by organisms to a manageable degree. In practice, this means measuring dozens of variables, and then presenting them as 2- or 3-dimensional graphs. Much of the technical challenge of phonetics concerns balancing the loss of information due to such a reduction against the ease of interpreting the resulting graphs. The method can be traced back to 1763 and Michel Adanson, in his Famille des Plantes, because of two shared basic principles, overall similarity and equal weighting, and modern phoneticists are sometimes termed neo-Adansonians. 2. Difference from cladistics. Phonetic analyses are unrooted, that is, they do not distinguish between plesiomorphies, traits that are inherited from an ancestor, and apomorphies, traits that evolved anew in one or several lineages. A common problem with phonetic analysis is that basal evolutionary grades, which retain many plesiomorphies compared to more advanced lineages, seem to be monophyletic. Phonetic analyses are also liable to be rendered inaccurate by convergent evolution and adaptive radiation. Cladistic methods attempt to solve those problems. Consider for example songbirds. These can be divided into two groups, Corvida, which retains ancient characteristics of phenotype and genotype, and Passerida, which has more modern traits. But only the latter are a group of closest relatives. The former are numerous independent and ancient lineages which are related about as distantly to each other as each single one of them is to the Passerida. For a phonetic analysis, the large degree of overall similarity found among the Corvida will make them seem to be monophyletic too, but their shared traits were present in the ancestors of all songbirds already. It is the loss of these ancestral traits rather than their presence that signifies which songbirds are more closely related to each other than to other songbirds. However, the requirement that taxa be monophyletic, rather than paraphyletic as for the case of the Corvida, is itself part of the cladistic method of taxonomy, not necessarily obeyed absolutely by other methods. The two methods are not mutually exclusive. There is no reason why, e.g., species identified using phonetics cannot subsequently be subjected to cladistic analysis, to determine their evolutionary relationships. Phonetic methods can also be superior to cladistics when only the distinctness of related taxa is important, as the computational requirements are less. 3. The history of phoneticism and cladism as rival taxonomic systems is analyzed in David Hull's 1988 book Science as a Process. 4. Current usage. Traditionally there was much debate between phoneticists and cladists, as both methods were proposed initially to resolve evolutionary relationships. One of the most noteworthy applications of phonetics were the DNA hybridization studies by Charles G. Sibley, John E. Ahlquist and Bert L. Monroe Jr., from which resulted the 1990 Sibley-Ahlquist taxonomy for birds. Controversial at its time, some of its findings e.g. the Galloncerae, have been vindicated, while others e.g. the all-inclusive, Sicaniformes, or the Corvida, have been rejected. However, with computers growing increasingly powerful and widespread, more refined cladistic algorithms became available which could test the suggestions of Willy Hennig. The results of cladistic analyses were proven superior to those of phonetic methods, at least for resolving phylogenies. Many systematists continue to use phonetic methods, particularly to address species-level questions. While a major goal of taxonomy remains describing the tree of life, the evolutionary relationships of all species, for fieldwork one needs to be able to separate one taxon from another. Classifying diverse groups of closely related organisms that differ very subtly is difficult using a cladistic method. Phonetics provides numerical methods for examining patterns of variation, allowing researchers to identify discrete groups that can be classified as species. Modern applications of phonetics are common for botany, and some examples can be found in most issues of the journal Systematic Botany. Indeed, due to the effects of horizontal gene transfer, polyploid complexes and other peculiarities of plant genomics, phonetic techniques of botany, though less informative altogether, may, for these special cases, be less prone to errors compared with cladistic analysis of DNA sequences. In addition, many of the techniques developed by phonetic taxonomists have been adopted and extended by community ecologists, due to a similar need to deal with large amounts of data. 5. Life history theory. Life history theory (LHT) is an analytical framework, one designed to study the diversity of life history strategies used by different organisms throughout the world, as well as the causes and results of the variation in their life cycles. 2. It is a theory of biological evolution that seeks to explain aspects of organisms' anatomy and behavior by reference to the way that their life histories, including their reproductive development and behaviors, post-reproductive behaviors, and lifespan length of time alive, have been shaped by natural selection. 2. It is a theory of biological evolution that seeks life history theory. Life history theory (LHT) is an analytical framework designed to study the diversity of life history strategies used by different organisms throughout the world, as well as the causes and results of the variation in their life cycles. 2. It is a theory of biological evolution that seeks to explain aspects of organisms' anatomy and behavior by reference to the way that their life histories, including their reproductive development and behaviors, post-reproductive behaviors, and lifespan length of time alive, have been shaped by natural selection. A life history strategy is the age and stage-specific patterns, and timing of events that make up an organism's life, such as birth, weaning, maturation, death, etc. 3. These events, notably juvenile development, age of sexual maturity, first reproduction, number of offspring and level of parental investment, senescence and death, depend on the physical and ecological environment of the organism. The theory was developed in the 1950s and is used to answer questions about topics such as organism size, age of maturation, number of offspring, lifespan, and many others. 5. In order to study these topics, life history strategies must be identified, and then models are constructed to study their effects. Finally, predictions about the importance and role of the strategies are made. 6. And these predictions are used to understand how evolution affects the ordering and length of life history events in an organism's life, particularly the lifespan and period of reproduction. 7. Life history theory draws on an evolutionary foundation, and studies the effects of natural selection on organisms, both throughout their lifetime and across generations. 8. It also uses measures of evolutionary fitness to determine if organisms are able to maximize or optimize this fitness. 9. By allocating resources to a range of different demands throughout the organism's life. 1. It serves as a method to investigate further the many layers of complexity of organisms and their worlds. 10. Organisms have evolved a great variety of life histories, from Pacific salmon, which produce thousands of eggs at one time and then die, to human beings, who produce a few offspring over the course of decades. The theory depends on principles of evolutionary biology and ecology and is widely used in other areas of science. Brief history of field. Life history theory is seen as a branch of evolutionary ecology, and is used in a variety of different fields. Beginning in the 1950s, mathematical analysis became an important aspect of research regarding LHT. 11. There are two main focuses that have developed over time, genetic and phenotypic. 10. But there has been a recent movement towards combining these two approaches. 11. Life cycle. All organisms follow a specific sequence in their development, 9. Beginning with gestation and ending with death, which is known as the life cycle. Events in between usually include birth, childhood, maturation, reproduction, and senescence, and together these comprise the life history strategy of that organism. 3. The major events in this life cycle are usually shaped by the demographic qualities of the organism. 2. Some are more obvious shifts than others, and may be marked by physical changes, for example, teeth erupting in young children. 8. Some events may have little variation between individuals in a species, such as length of gestation, but other events may show a lot of variation between individuals. 3. Such as age at first reproduction. Life cycles can be divided into two major stages, growth and reproduction. These two cannot take place at the same time, so once reproduction has begun, growth usually ends. 9. This shift is important because it can also affect other aspects of an organism's life, such as the organization of its group or its social interactions. 8. Each species has its own pattern and timing for these events, often known as its ontogeny, and the variety produced by this is what LHT studies. 12. Evolution then works upon these stages to ensure that an organism adapts to its environment. 5. For example, a human, between being born and reaching adulthood, will pass through an assortment of life stages, which include birth, infancy, weaning, childhood and growth, adolescence, sexual maturation, and reproduction. 3. 12. All of these are defined in a specific biological way, which is not necessarily the same as the way that they are commonly used. 12. Darwinian fitness. In the context of evolution, fitness is determined by how the organism is represented in the future. Genetically, a fit allele outcompetes its rivals over generations. Often, as a shorthand for natural selection, researchers only assess the number of descendants an organism produces over the course of its life. Then, the main elements are survivorship and reproductive rate. 5. This means that the organism's traits and genes are carried on into the next generation, and are presumed to contribute to evolutionary success. The process of adaptation contributes to this success by impacting rates of survival and reproduction, which in turn establishes an organism's level of Darwinian fitness. 5. In life history theory, evolution works on the life stages of particular species, e.g., length of juvenile period, but is also discussed for a single organism's functional, lifetime adaptation. In both cases, researchers assume adaptation, processes that establish fitness. 5. Traits. There are 7 traits that are traditionally recognized as important in life history theory. 4. 1. Size at birth. 2. Growth pattern. 3. Age and size at maturity. 4. Number, size, and sex ratio of offspring. 5. Age and size specific reproductive investments. 6. Age and size specific mortality schedules. 7. Length of life. The trait that is seen as the most important for any given organism is the one where a change in that trait creates the most significant difference in that organism's level of fitness. In this sense, an organism's fitness is determined by its changing life history traits. 6. The way in which evolutionary forces act on these life history traits serves to limit the genetic variability and heritability of the life history strategies. 4. Although there are still large varieties that exist in the world, Strategies. Combinations of these life history traits and life events create the life history strategies. As an example, Weinmiller and Rose, as cited by Lardillo and Delsic, propose 3 types of life history strategies in the fish they study: opportunistic, periodic, and equilibrium. 13. These types of strategies are defined by the body size of the fish, age at maturation, high or low survivorship, and the type of environment they are found in. A fish with a large body size, a late age of maturation, and low survivorship, found in a seasonal environment, would be classified as having a periodic life strategy. 13. The type of behaviors taking place during life events can also define life history strategies. For example, an exploitative life history strategy would be one where an organism benefits by using more resources than others, or by taking these resources from other organisms. 14. Characteristics. Life history characteristics are traits that affect the life table of an organism, and can be imagined as various investments in growth, reproduction, and survivorship. The goal of life history theory is to understand the variation in such life history strategies. This knowledge can be used to construct models to predict what kinds of traits will be favored in different environments. Without constraints, the highest fitness would belong to a Darwinian demon, a hypothetical organism for whom such trade-offs do not exist. The key to life history theory is that there are limited resources available, and focusing on only a few life history characteristics is necessary. Examples of some major life history characteristics include: age at first reproductive event, reproductive lifespan and aging, number and size of offspring. Variations in these characteristics reflect different allocations of an individual's resources, i.e., time, effort, and energy expenditure to competing life functions. For any given individual, available resources in any particular environment are finite. Time, effort, and energy used for one purpose diminishes the time, effort, and energy available for another. For example, birds with larger broods are unable to afford more prominent secondary sexual characteristics. 15. Life history characteristics will, in some cases, change according to the population density, since genotypes with the highest fitness at high population densities will not have the highest fitness at low population densities. 16. Other conditions, such as the stability of the environment, will lead to selection for certain life history traits. Experiments by Michael R. Rose and Brian Charlesworth showed that unstable environments select for flies with both shorter lifespans and higher fecundity. In unreliable conditions, it is better for an organism to breed early and abundantly than waste resources promoting its own survival. 17. Biological trade-offs also appear to characterize the life histories of viruses, including bacteriophages. 18. Reproductive value and costs of reproduction. Reproductive value models the trade-offs between reproduction, growth, and survivorship. An organism's reproductive value, RV, is defined as its expected contribution to the population through both current and future reproduction. Reproductive value models the trade-offs between reproduction, growth, and survivorship. An organism's reproductive value, RV, is defined as its expected contribution to the population through both current and future reproduction. 19. RV equals current reproduction plus residual reproductive value, RRV. The residual reproductive value represents an organism's future reproduction through its investment in growth and survivorship. The cost of reproduction hypothesis 20 predicts that higher investment in current reproduction hinders growth and survivorship and reduces future reproduction, while investments in growth will pay off with higher fecundity, number of offspring produced, and reproductive 18. Reproductive value and costs of reproduction. Reproductive value models the trade-offs between reproduction, growth, and survivorship. An organism's reproductive value, RV, is defined as its expected contribution to the population through both current and future reproduction. 19. RV equals current reproduction plus residual reproductive value, RRV. The residual reproductive value represents an organism's future reproduction through its investment in growth and survivorship. The cost of reproduction hypothesis 20 predicts that higher investment in current reproduction hinders growth and survivorship and reduces future reproduction, while investments in growth will pay off with higher fecundity, number of offspring produced, and reproductive episodes in the future. This cost of reproduction trade-off influences major life history characteristics. For example, a 2009 study by J. Creighton, N. Heflin, and M. Belk on burying beetles provided unconfounded support for the costs of reproduction. 21. The study found that beetles that had allocated too many resources to current reproduction also had the shortest lifespans. In their lifetimes, they also had the fewest reproductive events and offspring, reflecting how overinvestment in current reproduction lowers residual reproductive value. The related terminal investment hypothesis describes a shift to current reproduction with higher age. At early ages, RRV is typically high, and organisms should invest in growth to increase reproduction at a later age. As organisms age, this investment in growth gradually increases current reproduction. However, when an organism grows old and begins losing physiological function, mortality increases while fecundity decreases. This senescence shifts the reproduction trade-off towards current reproduction. The effects of aging and higher risk of death make current reproduction more favorable. The burying beetle study also supported the terminal investment hypothesis. The authors found beetles that bred later in life also had increased brood sizes, reflecting greater investment in those reproductive events. 22. RK selection theory. The selection pressures that determine the reproductive strategy, and therefore much of the life history, of an organism can be understood in terms of RK selection theory. The central trade-off to life history theory is the number of offspring versus the timing of reproduction. Organisms that are R-selected have a high growth rate R and tend to produce a high number of offspring with minimal parental care. Their lifespans also tend to be shorter. R-selected organisms are suited to life in an unstable environment, because they reproduce early and abundantly and allow for a low survival rate of offspring. K-selected organisms subsist near the carrying capacity of their environment K, produce a relatively low number of offspring over a longer span of time, and have high parental investment. They are more suited to life in a stable environment in which they can rely on a long lifespan and a low mortality rate that will allow them to reproduce multiple times with a high offspring survival rate. 23. Some organisms that are very R-selected are Semelparous, only reproducing once before they die. Semelparous